Hello, I am A.Kharchenko, and along with my colleagues H.Sidky, S.Michael, N.Hotaling, represent the National Center of Advanced Translational Sciences, National Institute of Health, Information Technology Research Branch, and X.Tal Informatics. I would like to tell you about our new feature extractor software, Nyxus. So the motivation of developments in the field of feature extraction is progress in microscopy techniques. Recent achievements in confocal and fluorescent microscopy and data acquisition equipment let us acquire massive amount of cell images of very high resolution. An example image matrix is on the left. Let's closer look at typical data high content analysis deals with. On the right, we see the whole bore microscopy image acquired in tiles. Individual tiles are on the left. They come from two acquisitional modes, recording data into separate channels. Nuclei channel, typically blue fluorescent DNA stain, and the cytoplast channel, the cell membranes. Not all the image content is informative. Essential part is taking place in cells, so you can see a cell segmentation mask on the center of the image. Segmentation is preceding feature extraction. A closer look into cytometry task performed. We need to run a large number of intensity, shape, neighborhood, etc. measurements of an intensity image within corresponding segmentation masks using the second image. The overlay shows a non-trivial intensity distribution within arrays, each having its own code. Non-informative background is black. Typical setup from the data perspective. We always work with a pair of intensity and segmentation mask images. Some segments lay nicely in a frame while some do so partially. Obviously, they continue on the mosaic tiles. All of these great alternative pieces of software are sufficient for us, for the HCA. No instrument meets all the requirements at the same time for speed, out-of-core processing scalability on large images, minimum set of comprehensive features, availability as user interface, Python library and command line interface, whole slide analysis mode, neighbor calculations, hierarchical relationships, usage of GPU, and more. So on this slide we see some quick benchmarks. Given the time-critical nature of HCA, its feature extraction needs to be efficient and sort of economical, providing as low cost of a single feature calculation as possible. This table illustrates Nyxus' leading position among alternative feature extractors, suitable for high-content analysis pipelines with respect to this benchmark. Nyxus is a fully open source software. It is deployable from the GitHub repository into any of the three targets, Python library, command line interface, and Docker image, covering three platforms, Linux, Apple OS X, and Windows. Nyxus uses other open source software, Active, and a couple of National Institute of Standards packages. Attempting to reach the performance of deep feature extraction and match other classical feature extractors, we implemented over 450 features: intensity statistics, image moments, texture features, morphology, radial distributions, bandpass filters, including Gabber, etc. All these features are runtime optional, meaning the user can selectively select any features she wants to have in the output. Purpose of this is saving compute time because each feature family has its own computational costs, obviously, and those times to compute costs are quite uneven. As we see, feature groups take expressively different time. So we design Nyxus architecture the way to perform calculations efficiently. Let's see what's under the hood of Nyxus. Workflow consists of scanning, accumulation, stitching, and feature estimation blocks. Each block implements a certain feature group with the intermediate results reused as much as possible. We use state-of-the-art open source TIFF file navigation software developed at National Institute of Standards. Its purpose is predictive prefetching mosaic image tiles while iterating pixels of previously fetched tile. So the feature calculation from input to output is a sequential process. Nevertheless, we managed to organize it in 3 blocks of logic, each block having its own parallel algorithm behind. Those blocks are displayed in 3 color zones. Zone number 1 is parallel when computer memory permits storing the whole pixel cloud of region of interest. Zone 2, the orange zone, is mostly sequential. Zone 3 calculates features in parallel batches by unique segment label. The middle zone operates in a sequential mode when a region of interest is so massive that its pixel cloud and temporary structures exceed the amount of computer RAM. An example is following a large region of interest of a neuron cell on the middle top image. Another important case is whole slide image feature extraction. Different datasets put different load on each zone of Nyxus architecture. Processing an image having 70 to 100 complex, relatively complex regions of interest results in increased morphology timing. We can see the increased erosions, for example, time share. Processing an image, processing a huge image having trivial sized and oversized regions of interest result in increased image scan timing. So the right hand side time structure, time breakdown is scanning some trivial segments and large segments that require following them one by one. So expectedly the scan is expanded up to 10% more. And finally, processing a large amount of simple shaped segments result in just decreased morphology timing compared to the first case. Some components of feature extraction timing make a significant contribution. Filters, image moments, and hair-like texture features. Apart from just running other less demanding features in parallel with any of these 3, the timing can be improved by using a GPU resource if it's available on the user's platform. Another point of active development is recognition of a hierarchical relationship between segments and calculating smarter, smarter aggregated features using knowledge about relationships. Also, we perform results validation through image by micro standardization initiative. Availability of a rich classical feature set lets one create deep learning pipelines without extracting deep features. The benefit of a classical feature is that it's typically human design, intuitive, easy to validate, requires less computational resource, and can be very specialized for an application. Our colleagues Hamda Abbasi and Nyxus set up a training pipeline to perform an accuracy test comparing features provided by Nyxus compared to ones provided by Deep VGG16net. On this slide, we see a pretty typical pipeline, beginning with feature extraction, then outlier detection, feature processing, and selection, and finally training a PyTorch implementation of a tabnet classifier. The goal of this test was predicting a tissue type. The test result, along with a confusion matrix, is presented on this slide. A matrix per each feature extraction situation. The Nyxus-based pipeline outperformed the deep feature-based one in predicting this 66-class tissue type by actually a large margin. In the summary, so we developed Nyxus, an open source optimized C++ based feature calculator, providing CLI, Python library, Docker image for 3 most popular software platforms, featuring nested arrays, very rich feature set, and actively expanding in as a Python interface and GPU. Please meet our group at the National Center for Advancing Translational Sciences.